justify that much insulin. And if you don't, then you could crash. Okay? Scary. Some places, what do they do to help you out of something like that? Have you drink
Sugar water?
orange juice? Orange juice, they would. Sugar water would be sort of the same thing. Okay?
It's kind of the same thing they do for when you donate blood, right?
I'm not sure what they have you do. They give you cookies.
Yeah, they use something with sugar.
Yeah.
And they give you like little drinks.
Oh, Gatorade,
Something like that.
okay. So they want to kind of build your sugar up again because they took blood out of you. Yeah. Hemoglobin. All right. Let's see where we want to go next. I have an idea. That was good. Let's see what else we can do. See, that was chapter 2. I jump in and out of these. We have nice pictures, some in one, some in the other. Well, let's look at this. Why not? What are we looking at? It's page 92.
Nucleus of the cell.
Nucleus of a cell. Do you see the nuclear membrane? Do you? It's not a nuclear membrane. It's a nuclear envelope because it's a double membrane. So the nuclear envelope goes around the nucleus and it's a nuclear envelope, double membrane. And you got holes called nuclear forks. And you got fuzzy stuff in here called chromatin. Chromatin is what you get when you pack it together, you get chromosomes. But you only have chromosomes when you're doing the dance of the chromosomes, mitosis, that's right. Okay, what's the little sphere in a sphere? It's the nucleolus, okay? And this is actually connected here at the nuclear envelope with this bluish stuff with dots on it. Who is this? Rough endoplasmic reticulum, and it has cisterns. What's that sound like? Sarcoplasmic reticulum. So you see, sarcoplasmic reticulum is actually an offshoot of this. It's actually a modification of this. Okay? And sarcoplasmic reticulum stores what inside? Calcium ions. But that's not what these red dots are. What are these? Like fleas on a dog, jumping on and off. What are these? Ribosomes. Yep, they actually can jump off and on. Don't ask me exactly how they do that. I don't know, but they do it. And what do they do, ribosomes?
Make protein. Help.
They do. They make proteins. What would be your technical way of saying that? Protein synthesis. That's right. Okay. Tell me what that process probably ends up being. Protein synthesis, joining amino acids together. What do you think's coming off? Yeah, that's not coming off. What's coming off? Water's coming off. Dehydration, synthesis. That sounds reasonable. Okay. So you're used to thinking of chromosomes like this, the black X, but it's really not around except for the dance of the chromosomes. The rest of the time, what's it like? Long, wispy threads of chromatin.
What is, so the chromatin isn't even in the nucleolus, it's in the.
No, it's out here in the rest of this. Yeah. Okay. What is it?
What is it?
Well, I'm going to tell you about it. I'm not ready to do that yet. Okay. Let's go a little further. Did we look at this? No, we did not. What would you call this? A double helix. Who's the double helix? DNA. And what are the steps? Nitrogenous base pairs. What are the handrails? Alternating. deoxyribose, it has to be that, it can't be ribose. This is DNA. Deoxyribose alternates with phosphate. That's right. And have you ever had a piece of string or something like that, maybe even yarn, and you just didn't really want to throw it away? So what'd you do with it? You can wrap it around a pencil. You can wrap it around a spool if you have one. So what are we gonna wrap our long DNA molecule around? Little stones. See little stones? Like little pebbles, little stones. They're actually called histones. And what are they? Another form of proteins. These are proteins. So you wrap the DNA around the histones and you get them in little groups called nucleosomes. And you say, what do they look like? Well, there is an actual picture. This is small. How did they get this picture?
Probably
Electron microscope. Notice it's not colored. You don't get colors with electron microscope. You get blacks and whites and grays. Because what are they using instead of light? Electrons. Electrons. So you're not going to have the colors. So as we keep on going down here, this gets twisted and twisted and twisted, which is meant to show you that's what the X chromosome, well, X type of chromosome, any X chromosome, I'm not saying X and Y, I'm just saying these X's, these black X's, that's really of what they're composed, which is fascinating. Okay. Because they were trying to spell it out for you there, how it is, what it is. Okay. So that was neat. Let's see what else we can talk about right now. We didn't talk about this, so we want to talk about it. You see the potatoes? Not black olives, potatoes. Who are they? Enzymes. The ace that they place at the end here is telling you this is an enzyme. DNA polymerase. So what are you making? DNA. You're stretching out this molecule by making a new half. And this is the same kind of potato, but it's going the opposite way. So it has a harder time doing that. All right. Well, first of all, you had the ladder, and you split it apart with another potato. So the enzyme unwinds your ladder, so you have two halves of the ladder. And now the potatoes here, DNA polymerase, wants to make a new half. If you have to make a new half of the DNA ladder, you need a lot of nucleotides. And here they are. The cell just goes ahead and makes these. So these are around. There's no problem. There's no shortage. You don't run out and have to go to Lowe's or Home Depot or Walmart or someplace to get more. They're around, and the potato says, I'm so glad because I've got to see that these go in the right place, because we know that the red one always goes with, no, not the red one. Red one goes with the blue one. Okay. So you can color, you've got the color code. They're not really color coded like this, but it helps our discussion to think about it. Okay. And so you just keep putting these in, and you got a new half of the strand, keep forming. Okay. Is this hard to do? No. Zip, going across here. This one gets jealous. It's the same kind of enzyme, but it says, look, I'm having to do the hard work because I'm going this direction. I can't go zip. I have to make pieces and glue them together. So this one doesn't grow as quickly. So it's our lagging strand. Whereas the other one is just zip. That's our leading strand, the one that forms the easier way. Leading strand, lagging strand. But all in all, it's just fine, because when you're finally done, you're going to have two ladders, not one, and they're going to be absolutely identical to the original. So what are you sticking in here, thumbtacks or what? Nucleotides. Now tell me, what makes up a nucleotide? Nitrogenous base, phosphate, and what kind? Pentose sugar, good. Okay, so that's a nucleotide. The cell made these, you can just stick them in where you need to. I think they look like thumbtacks, so that's what I call them. Not all of us this time. Okay. What are you doing next? DNA replication is what it's called. If I didn't clarify that, I want to. This is DNA replication, which just means making more DNA. You ever watch the science fiction shows and they have a replicator and they're out in space and they say, I'd like a hot fudge sundae, and voila, there it is. And you say, oh, I can't wait for that day. Okay, well, you don't have to, because I don't think they do that yet. They're probably working on it though. I can't say they're not. Yeah, well, I'm not sure I'd eat that. They're printing up all, somebody said they 3D printed an ear. Yeah. They said a kidney too, but I can't see how it works. Yeah.
Oh, have you not seen how they do that? It's in the experimental stage now, but they have this, they did it with a heart, and they put this stuff on it that keeps the collagen, so the structure is there.
So it's still there.
It's like a ghostly white
Ghostly,
heart, and then they seed it with the cells of the donor.
Oh, they actually do
And
see
some
it.
stem cells, and it grows into a heart on the scaffolding.
Have they used it yet?
I don't think they've used it on an implant, but they're just doing the.
They're getting there.
Yeah. They've been using pig hearts, cause that thing's so dumb.
That's not a bad idea, because we only have so many hearts.
And you'd never be rejected because it's your own.
Exactly. If it's your own.
We just have an insurance policy where there's a warehouse full of organs.
Yeah, I like it. It has potential. Do you want a horror story? No, you don't want a horror story.
I do.
A horror story?
Yeah. Yeah, you can't just like.
This was real.
Yeah.
This was real. You know, they replace valves in the heart, a valve replacement, with artificial ones. Now, they used to do pigs and cow, especially pig valves, they would put in, but they have artificial ones. And they ended up with a recall.
For the artificial ones?
Uh-huh. Which meant we were gonna have to have it taken out.
With no backup.
Well, they could put a new artificial one in that wasn't defective, it was defective,
Okay.
which was bad
Yeah.
because it could just stop.
Just drop.
Now what are you gonna do? Say, I'll just wait it out? Oh, yeah, I don't think so. So you would be very unhappy, but you'd be getting a new one. Or a pig one, if you could. But that was, I mean, car getting recalled, that's one thing. Heart getting a recall, heart valve, so that was no good. That happened. All right, so, okay, oh, wow, I got that faster than I wanted. Okay. So here we go. I'm gonna interview the cell. Okay, Mr. Cell, I can be Mrs. Cell, I don't care. I'm gonna ask you about your life. And the cell says, well, that was very nice of you. My life cycle as a cell. Well, I spend, first of all, my first part is in this green area called interphase. But if I want to do the dance of the chromosomes, I need to move into the mitotic phase. And what's the first part of mitotic phase? Mitosis. Can you name the parts? Prophase, metaphase, anaphase, telo or telophase. And then you say, is there anything else in this mitotic phase? Yes, the cell finally splits apart. That's called cytokinesis. Cytokinesis. What do you have when you're done? Two daughter cells, just like the original cell. And so what's the original cell? What are the daughters going to do? They're going to go back in interphase. Now you have some cells that don't want to do the dance. They don't want to do the mitotic phase because they're too complicated, it's too hard, they don't wanna do it. Brain cells. So they're working on this. They're working on different things right now, some exciting stuff from what it sounds like, okay? But the more difficult things, they resist change. Because who wants to mess up their brain cells? You don't really want to do that, okay? But that's interesting. So, if you're gonna split in two, don't you need double everything? Don't you need double chromosomes? And what are in the chromosomes? The DNA. So you have to do DNA replication, that's right. So when would you do that? During mitosis? No, when would you do it? In interphase. Which part of interphase? The S growth and DNA synthesis phase, the S phase. S phase of interphase is when you do DNA replication, right. Did you catch that?
Can you repeat that one more time?
Did that help? That wasn't what you wanted. Okay. When you are going to do DNA replication, you do it during interphase, the S subphase. S subphase.
So we can format that, you say like S phase of interphase.
S phase or S subphase, yeah. That's when you do it. Now, have you ever heard people say that mitosis is part of interphase? Or that interphase is part of mitosis? No, it's not. You can see that now. But I'll ask questions to students, and they'll be trying to tell me interphase is part of mitosis, and it's not. Here's mitosis. This is pages 100 and 101. I'm not going to talk about that right now, but those are pretty pictures. But I do want to talk about this. If I can, let's try this. Let's try doing that. No. There, that's better. All right. These are really nice pictures. Imagine a book taking the time to put these colored pictures in it and pay the cost. They must think it's worth it, and it is. What's the first thing you do when you look at a picture? Get oriented. What's this purple sphere down here? Nucleus. Nucleus. What's the boundary called? Nuclear envelope with nuclear pores, so things can go out. Okay. In here, this is on page 106. On this one, I see the DNA ladder in there, and I wished I had it there. Maybe I can move it. See the DNA ladder? The twisted ladder shape, double helix. Okay, what are the steps? Nitrogenous-based pairs. And the pairs are held together by weak hydrogen bonds. Good. So you can zip and unzip the DNA strands. Okay. Why did we, okay, no, I won't ask that. All right. If you have it unzipped, then you can use this path of the strand and build a new strand on it, but it could be RNA. And then you can rip off the RNA and throw it out a hole. Okay? So you had this unzipped so you could build the new RNA and throw it out the hole. And I like to call it the meat. That RNA is the meat, because it's messenger RNA, and and you throw it out, and guess who is paying attention and comes and makes the sandwich?
Right.
The ribosome. You see, ribosomes, you think of them as little dots, but they're actually two halves of a bun. See, there's a little bigger half and a little smaller half, and they float around as the two halves. But when the meat comes, they grab it like a bun and make a sandwich. So the messenger RNA is surrounded by the bun there, who's going to slide along the meat. Okay, well, let's go over here to the other page. This is page 107, right? 107. So there's some action going on here. This is kind of interesting. So remember, you've got the bun sliding along the meat, and now we get some guitars. The guitars come in and land on the meat like flies, but only if they're permitted to do that. So what are the guitars carrying? The top of the guitar, what's there? An amino acid. And it's not just any amino acid. It's the only one this guitar is allowed to carry. So guitars are snooty about this. They can only carry one kind of amino acid. If you need it for another kind, you gotta look for another guitar. Because they only carry their own special amino acid type. So if they've got the right one, and conditions are right, they can land on the A site of the bun. It's called the A site. It's actually on the meat. Well, let's look at the bottom of the guitar. I have three what? They're not nucleotides now. They're just nitrogenous bases. Yeah. Who are they here? G, A, and U. What are you dealing with if it's a U? RNA, that's right. Okay, what's the bottom of the guitar thing called? The anticodon. The anticodon is here. It wants to land, if permitted, on the meat, these three nitrogenous bases, which are actually called the codon. Anticodon, codon. Okay, if it works, this one was a G, what does it have to match up with?
C.
The C. Oh, it does. Here's an A, what does it have to add a match up with? U, because this is RNA. If it weren't, it'd be a T. And the U has to go to an A. Well, look, you're cleared for landing. Go ahead and land. All right. Which site are you landing on?
On the bun side?
A site. A site. And the bun keeps sliding, so the guitar landed on the A site, but now it's on the P side and gives its amino acid to the long chain of pearls, long chain of amino acid. It just puts it on. Who was the last one to put one on here? Somebody with, well, this is it, with proline. Okay, but now leucine wants to add to the chain, so it's coming out, landing, getting over there, giving off its amino acid. Now it says, I'm in the E site. E for empty, or E for empty was mine, exit. So the guitar says, I'm empty, I'm gonna leave, takes off, where's it go? Why, it goes to the moon, of course. You didn't know that? It goes to the moon. And did you know the moon is made of blue cheese?